Now with this, let's see why does a neuron die. So we understand that the neuron dies, and so just a quick morphology here. In the, let me just write it down, morphology, appearance of the substantia nigra or the midbrain. Morphology, macroscopic and microscopic, and they both are step questions. Macroscopic morphology is that the substantia nigra, pars compacta plus locus coeruleus, they look pale. They look pale. Why do they look pale? Because there is a degeneration of the fibers which are dopaminergic and melanin in nature. So because of that degeneration, the substantia nigra, pars compacta and the locus coeruleus look pale. That is in microscopic with the bare eyes. Microscopic, what they see is, so if this is a neuron, so this is the axon and these are the dendroids. This is the neuron cell body. What they see microscopically is the accumulation of Lewy bodies. Lewy bodies are pink colored accumulations inside the cell bodies and even at the junctions of the dendrites, which blocks the neurotransmitter release. And what happens is these accumulation, the Lewy body itself looks like this. It is more darker in the center and then slightly lighter on the sides. It is made up of the clumps of the alpha-synuclein protein forms into the beta-pleated sheets or clumps or aggregates. Just like in Huntington's protein forms a clumps, here the alpha-synuclein forms a clumps. Just like in Alzheimer, tau proteins form the clumps. These clumps accumulate in the form of the Lewy bodies. In the center, the clumps are really rigid and dense, and then the side clumps are lesser and open. So they are probably accumulating on the side, and so this is more open area. Now that is what is the microscopic view. How does this happen? How does alpha-synuclein appear and start making the clump? So let's look at that process here as well. So what happens is this. Alpha-synuclein, and there are multiple theories. This is an area of active research, so I cannot have you do PhD over here, but important thing for you over here is to understand from a steps point of view, what are the questions that you need to answer. Number one, from steps point of view, you need to understand that there is Lewy bodies present. Number two, what is the role of the alpha-synuclein? How does that happen? So what happens is this. Let's say if this is the alpha-synuclein protein, I'm just making up a protein. What happens is that there is a chemical, there is a change in the gene, there is a mutation in the gene. That mutation results in the polarity change in some pieces, and actually just one nucleotide can cause this problem. When the polarity changes, then multiple proteins, they clump up together. And you know that they can go from the primary structure to the secondary structure. That is what proteins do, right? So primary structure, secondary structure, and tertiary structure. So in this case, they go to the secondary structure of the beta-pleated sheet. So the normally alpha-synuclein is a coiled-ish, but it's straight coil-like thing. When multiple coils come together and they start bonding together, they end up making beta-pleated sheets. Those sheets are the ones that start accumulating and aggregating with each other to make fibrils, not neurofibrils, just the fibrils. These fibrils are the ones that are making the Lewy bodies. Now the question is, how do these fibrils cause problem? So there are multiple hypotheses. I'll just go to the two hypotheses over here. One hypothesis is that this aggregate, the Lewy body, this is important, please remember this one. This aggregate captures some proteins with it which are necessary to inhibit the ubiquitin proteins. And you know that the ubiquitin protein is necessary, ubiquitin is necessary for apoptosis or cell death, automatic cell death, or the suicide by the cell. So now what happens is, when those fibrils of the alpha-synuclein also trap some proteins that were inhibiting the ubiquitin, what is going to happen? Ubiquitin is now free to do its function. What is the function of ubiquitin? Apoptosis. So because of increased alpha-synuclein, ubiquitin becomes freed up. That freed up ubiquitin starts a process of apoptosis. That is one of the theories. Other theories are that these nuclei, these aggregates, create the physical problems inside the cell. They accumulate there, they disrupt the function of the antioxidants, they disrupt the function of the mitochondria, they disrupt the neurotransmission inside the neurons, and the cell starts functioning less. End result of this is that the dopaminergic cells are more commonly affected and they die. Now if you come back here, knowing this, dopaminergic cells, here is the D2 pathway. This is the D2 pathway. Now just like as I said before, D2 pathway here, if that is affected, it can actually be affected in other areas too. Cool. So this is the basic problem in the Parkinson's. The end result of this is that once the substantia nigra pars compacta starts degenerating, once the inhibition or modulation on the neostriatum starts going away, thalamus becomes disinhibited and the rigidity starts appearing.
Guys, thank you very much for watching. Make sure that you like, subscribe, and share this video. Like it if you like it. If you don't like it, then don't like it. And then subscribe if you want more videos. We upload videos regularly, so if you subscribe to the channel, you can get a notification and the video will appear in your inbox. And if you hit the bell button as well, then you can get the notifications for this as well.